the DNA, right? The DNA we are going to discuss in detail in this particular chapter, and how the RNA is formed from the DNA. Like this, we are going to discuss one by one in detail, right? So the structure of the polynucleotide. The structure of the polynucleotide indirectly indicates the cluster of nucleotides. Cluster of nucleotides. So cluster of nucleotides in the sense which is going to determine this is the way the particular organism expression should supposed to be. So the nucleic acid is made up of a specific set of nucleotides. The nucleic acid is made up of a specific set of nucleotides. Nucleic acid is made up of specific set of nucleotides. This nucleotide is responsible for determining character. This nucleic acid is the part of the DNA. The DNA is a part of the chromosome. DNA is a part of the chromosome. So the chromosome is having the DNA. The DNA is having the nucleotide. The nucleotide is controlling the RNA formation. In which way the RNA should be formed. In which way the protein should be formed. The protein formation is controlled by the RNA. RNA formation is controlled by the DNA. This entire sequence we used to call as gene expression. This is the way the gene is getting expressed. Gene expression is also called as central dogma. Gene expression or central dogma denotes the protein formation from the DNA. Okay, or else we can say the RNA formed from DNA is making the protein. In any way, we can say all the grounds we discussed with you. Now we are coming for the next phase of explanation. We know very well the DNA structure is bringing the characters for many generations. This is the way the shape of the organism should supposed to be. This is the way the size of the organism should supposed to be. We know very well the macrophage symbol. Okay, they given a psi into 174. The particular macrophage has a DNA molecule consisting of these many number of nucleotides. These many number of nucleotides. The macrophage has DNA molecule consists of these many number of nucleotides. Okay, so that means the smallest microorganism of this world is a macrophage, which is nothing but a virus. We all know very well virus is a structure which infects the bacteria. So virus is a microorganism that infects the bacteria. We all know very well, right? So this macrophage is a virus that infects the bacteria. In this macrophage, the DNA length is about 5386. These three number of nucleotides are present in the DNA, right? So now if you're coming for the macrophage, so the DNA of the lambda macrophage is more extensive with 48,502 base pair. So the lambda macrophage is having a specific DNA. The DNA is having these many number of nucleotides. Okay, the nucleotides is having the base pair. So I am saying base pair, I am saying about the base pair of the nucleotide, I am saying base pair of the DNA, everything is same. So the macrophage DNA is having this many number of base pair. Okay, this many number of nucleotide. Instead of saying 5386 nucleotide, okay, I can't say these many number of base pairs are there, right? So you should be very careful in saying all these messages. Okay, so in the Escherichia coli, these many number of base pair 4.6 into 10 to the power of 6 base pairs are there, right? So the haploid content of the human DNA, if you're talking about the haploid content of the human DNA, is having half of the parental DNA. Half of the parental DNA. Who is having half of the parental DNA? The haploid content of the human DNA indirectly speaks about gametes. Gametes are nothing but sex cells or reproductive cells. So the sex cells or reproductive cells are having haploid number of chromosome, which is nothing but half of the parental chromosome, which is symbolized with single N. All right, this is the way you need to remember. What do you mean about haploid content? How I can symbolize single N? Half of the parental chromosome. Half of the parental chromosome. So half of the parental chromosome. So half of the parental chromosome. So half of the parental chromosome is present in the haploid cells of the human, right? The haploid cells of the human indirectly speaks about sex cells. Either you say sex cells or gametes or reproductive cell. Okay, that is having half of the parental chromosome, right? So now we need to understand the next basic concept in the structure of the polynucleotide. I already told you it is a many nucleotide. Okay, this polynucleotide is a part of the DNA. DNA is a part of the chromosome. So the structure of the polynucleotide if you are seeing this information, um, can you see this? The structure, how it is designed, okay, in a periodical order. Okay, we studied this DNA structure in the grade 11 in the biomolecule chapter, right? In the grade 11 biomolecule chapter, we saw this already, right? So can you see this? This adenine is getting attached with the guanine. So adenine is getting attached with the guanine, and guanine is getting attached with the thymine. So whenever we are narrating like this, we should not supposed to use base pairing. Why? Because this entire structure indicates only one DNA strand. This entire structure represents only one DNA strand. We should not supposed to use the word adenine is base pairing with the guanine. Guanine is base pairing with the thymine. Like this, you should not supposed to say, right? So in the yesterday's session, when we taught the principle of inheritance, at the time I told the adenine, guanine, cytosine, thymine in the different order is determining a character, right? So this adenine, guanine, cytosine, thymine in the different order is determining character, right? So, and after the adenine in a single strand, anyone can come. After adenine in a single strand, anyone can come. Okay, but in the opposite strand, obviously only thymine can come. Only thymine can come. Only thymine can come next to the adenine in the opposite strand for base pairing. Be very, very careful when we are saying the pairing between the two nucleotide of the DNA. For example, this strand is one DNA, other strand is one DNA. So two strands are a separate DNA which is having the base pairing connectivity. Okay, the base pairing connectivity possibility uh, is aligned in this way. That means what? A can base pair only the thymine and guanine can base pair only the cytosine. Adenine can base pair with thymine, guanine can base pair with cytosine. Adenine can base pair with thymine, guanine can base pair with cytosine. Are you getting my point or not? So this is the way you need to understand the message, right? So see this structure, adenine. After adenine, anything can come. There is no rule. But in the opposite stand, for joining the adenine, only I need to have the thymine. So adenine thymine always establishes double bond, whereas the guanine and cytosine establishes triple bond. This double bond and triple bond always indicates hydrogen bond. Like this in the board exam, they will ask. Name the bond which is present between the A and T and G and C. There is nothing but a hydrogen bond. Also, we can call as van der Waals forces. So van der Waals forces, we need to understand these van der Waals forces is a type of hydrogen bond. This hydrogen bond is present between the adenine and thymine as a double bond, between the guanine and cytosine as a triple bond. Okay. Now, if you're talking about a single nucleotide, beside having nitrogen base, it also has pentose sugar with the phosphoric acid or phosphate group. In the CBSE board exam, the nucleotide consists of what? Whenever they're asking, we need to say the nitrogen base might be a purine or pyrimidine, next to pentose sugar, next to phosphate in the form of phosphoric acid. Either you say phosphate group or phosphoric acid. So phosphate is also called as what? Phosphoric acid. Again, back I'm repeating, phosphate is also called as phosphoric acid. So phosphate is also called as phosphoric acid. Are you getting my point or not? So phosphate is also called as phosphoric acid. Like this, I can't say everywhere. In the DNA, I am pinpointing the phosphate as phosphoric acid, which is having the potential to attach with the other nucleotide in a same strand, but not between the two different strands. So the phosphoric acid is a connectivity between the two nucleotide of the same strand, but not between the two nucleotide of the different strand. Be very careful when we are narrating the phosphoric acid linkage between the two nucleotide, right? So now the nucleotide is totally different from nucleoside. Be very careful. This nucleoside is not having the phosphoric acid or phosphate, whereas nucleotide has phosphoric acid. The nucleotide linkage through phosphodiester bond results in the formation of polynucleotide chain. Polynucleotide chain indicates what? One nucleotide is interconnected with the other nucleotide. So one nucleotide connectivity. One nucleotide is getting connected to the other nucleotide. One nucleotide is getting connected to the other nucleotide. One nucleotide is getting connected with the other nucleotide. So what we need to understand is one nucleotide is connected with the other nucleotide because of the phosphodiester bond. Phosphodiester bond. But between the two nucleotide of the two different strand, between the two nucleotide of the two different strand, phosphodiester bond is not present. I already told only hydrogen bond is present. The hydrogen bond, if it is A and T, double bond. If it is G and C, if it is G and C, triple bond. If it is G and C, triple bond, right? So if it is G and C, triple bond. If it is A and T, double bond, right? So just randomly, what all the bonds, covalent bond, ionic bond, okay, coordinate bond, metallic bond, all the bonds we should not supposed to write. So then they will make you to get less mark. So be very careful. So between the one nucleotide, another nucleotide, or the two different strand, what particular bond is present? Phosphodiester bond is present like this. We already told you, right? So now we are coming for the next major session, historical background. In the historical background, who discovered the DNA for the first time in the world? The nucleon is a scientist? No. Friedrich Miescher is a scientist? Yes. What do you mean about nucleon? It's a Greek word. What it says about? It says about a primitive nucleus material. A primitive nucleus material is called nuclein, right? We need to understand basically the DNA structure faced the technical challenges in 1953. Okay, here we know very well in the ribose sugar there is a free hydroxyl group in the second carbon atom. There is a free hydroxyl group in the second carbon atom. Furthermore, in the RNA, uracil is found in the place of thymine. So with the uracil being a chemical analog of the thymine, right? So chemical analog means similar chemical structure. Who is having? Uracil is having. Thymine is having. Thymine and uracil structure not exactly similar, more or less similar. There is a lot of difference between exactly similar, more or less similar. So uracil and thymine are more or less similar. Okay, and second thing is the uracil you can find only in the RNA, whereas thymine you can see only in the case of DNA. The uracil we can see only in the case of RNA. Only in the case of RNA, right? Uracil we can find only in the case of RNA. So we're talking about, again back I'm repeating, uracil we can see only in the case of RNA. Okay, thymine we can see only in the case of DNA. So what we need to understand basically, the Francis Crick and Watson are the two great scientists discovered the double helical structure of the DNA, right? By using X-ray diffraction studies, okay, given by this Maurice Wilkins and Rosalind Franklin. So Rosalind Franklin and Maurice Wilkins, okay, and given a beautiful X-ray diffraction study of the DNA. And based on this, the DNA helical structure was established properly at the more valid bond linkage information. Okay, who worked on this? Watson worked on this, right? So what we need to understand basically, the X-ray diffraction means in the sense, basically for this particular sentence, a lot of hidden sentences are there. That is not there in the slide. I need to narrate. Calmly you listen to me. DNA after purifying and crystallizing at very low temperature. DNA after purifying and crystallizing at very low temperature. We will be getting a structure called lattice structure. This lattice structure is a structure which is having the efficiency to do the diffraction. Lattice structure is having the efficiency to do the diffraction. So diffraction means, you know very well, the X-ray after falling over the lattice structure of the DNA takes the image of the DNA crystals. The DNA crystal image is taken by the X-ray which is falling over this particular DNA crystals, right? So now what else we need to understand basically is, we know very well the double helical structure of the DNA, okay, was discovered by Watson and Crick. For that they got the Nobel Prize. We know very well this X-ray diffraction study was not given by Watson and Crick, okay? So I think so you understood this message, and I already told to you the RNA was dominant when we are not there before 5 million years back, before 10 million years back. At the time, what happened? Most of the species are having RNA as a genetic material. Most of the species had RNA as a genetic material. That RNA is getting slowly tuned to become DNA after getting mutation, because RNA has higher probability of getting mutated than the DNA because of the free hydroxyl group in the second carbon atom. So because of the free hydroxyl group of the second carbon atom of the nucleotide, okay, the RNA is highly vulnerable or susceptible for getting mutated. The RNA is highly vulnerable. The RNA is highly vulnerable for getting mutated. Okay, again, like I'm repeating, RNA is highly vulnerable or highly prone for getting mutated. Why? Because it is having free hydroxyl group, unlike DNA. So the free hydroxyl group of the ribose sugar makes the DNA, not makes the DNA, makes RNA to get mutated very faster. DNA also undergoes mutation, but very slowly, Okay, than the RNA, right? So what we need to understand basically the importance of the 2' hydroxyl group of the second carbon atom of the ribose sugar of the RNA. Like this, we need to understand. In the RNA, basically, again, back I'm repeating, in the RNA, ribose sugar is present. In the ribose sugar, free hydroxyl group is present. So in the RNA, ribose sugar is present. Okay, ribose sugar is having what? Free hydroxyl group in the second carbon atom. So the free hydroxyl group of the second carbon atom is highly prone for getting water. Is highly prone for getting water mutated. Is highly prone for getting mutated. That is the message we need to understand, right? So now, this is the entire structure is a double helical structure of the DNA. Can you see how the double helix is coiled? And then the distance between the one pitch and other pitch. The distance between the one pitch and the other pitch. Can you see this? 3.4 nanometer. 3.4 nanometer. Right. So now we are entering into the next important concept is nothing but genetic implication. So complementary base pairs allows the prediction of one DNA strand sequence from the other. So here during DNA replication, each strand acts as a template leading to the formation of the identical daughter cells. So this double-stranded DNA, once it is going for replication, it get assorted from each other, so that the daughter DNA will be formed by using the template or parental DNA. By using the template or parental DNA. By using the template or the parental DNA. By using the template or the parental DNA. So by using the template or the parental DNA, again back I'm repeating, by using the template or the parental DNA, that I given as a dot one, template or parental DNA, the new daughter strand I written in the light red. So again back I'm repeating, new daughter DNA strand. New daughter DNA strand. So new daughter DNA strand. So new daughter DNA strand is a strand which is formed by using the template DNA or parental DNA. Right? So now what we need to understand is the central dogma is nothing but the replication process where the DNA is synthesized, transcription process where the RNA is synthesized, and translation is the process um, where the protein synthesis happens, right? So DNA synthesis and RNA synthesis and protein synthesis, all these things together is called as central dogma, also called as gene expression. Okay. Now we know very well the DNA strands runs anti-parallel to each other. So anti-parallel to each other in the sense what we know very well the if one DNA strand is running from 5' to 3', 5' to 3', other DNA strands runs from 3' to 5', 3' to 5'. So it's always in the double-stranded DNA structure. So if one strand is running from 5' to 3', other strand runs from 3' to 5'. Why it is called 5'? Why it is called 3'? We told already n number of time because of the free hydroxyl group in the fifth carbon atom. Because of the free hydroxyl group in the third carbon atom of the nucleotide. So free hydroxyl group gives a name. Again, back I'm repeating free hydroxyl group. Again, back I'm repeating free hydroxyl group. So free hydroxyl group of the nucleotide present at the edges of the DNA is given the name 5' and 3'. Right? So now the two DNA strands are running anti-parallel. So if one strand is running in this direction, the other strand is running in the other direction. So this we used to call as anti-parallel running of the DNA. All right. Now we are coming for the next point is if you're talking about right-hand coil with a pitch of 3.4 nanometer. So pitch of 3.4 nanometer and 10 base pair will be there per the turn, which is not applicable for other than human. Only in the case of human DNA. Only in the case of human DNA. Okay, one turn is having 10 base pair. One turn is having 10 base pair. One turn is having 10 base pair. Are you getting my point? So now, if you're talking about the next major concept, the other secondary structure of the DNA, which is nothing but the packed structure. Before that, we'll discuss some important examples, questions, etc. Right? So now let me start discussing about the questions. This question is exactly taken from 2023. Okay, can you see this blaze here? Okay, this question is taken exactly from 2023. So identify the region X, Y, and Z involved in the process of transcription. Okay, as shown in the systematic representation. Right. So we know very well the sigma factor is not a terminator. Sigma factor, factor Y. Okay, always starts the initiation process. Always starts the initiation process. So obviously, what the C option will be the correct answer. Can you see this? C promote X promoter, Y sigma factor, and then the Z denotes what exactly the RNA polymerase. So the option C is the correct answer. So what we need to understand basically by having repeated practice, we can understand sigma factor starts the RNA synthesis or initiates the transcription first phase. Transcription first phase is initiated. Transcription is started. Okay, here in this occasion, the sigma factor is very, very important. Okay, for the RNA synthesis to happen. For the RNA synthesis to happen, for that, we already given one question narration of the previous year's paper. Okay, now we are coming for the next in the molecular basis itself. Okay, in the DNA fingerprinting. Calmly, if you're listening, okay, what is the function of the DNA fingerprinting? By doing DNA fingerprinting, I can find out the issues in the child dispute case. In the case of a rape case, in the case of murder case, I can find out the consent culprit. Okay, and then I can correlate with the victim samples, and then we can take those set of people to the jail if they would have committed the mistake, right? So this is 2023 paper, right? So any one of you are having any doubt in this junction, please let me know. So in this, which band is more helpful? Can you see this? The band 1 and 3. C is the correct answer. So with the child X indicating a what a potential percentage, right? 1 and 3 is showing maximum matching, right? So the 1 and 3 or the parent 1 and 3. Okay, now we are coming further. So how you chosen 1 and 3 are having the maximum probability? Why not the 2? Can you tell me the reason? Like this, whenever you're saying, can you see this? The DNA structure which is there in the band 1 area. Okay, it is exactly coming here. This is coming here, and then this is coming here. So this is coming here. So this is coming here, right? So all these things are coming this way. So what I'm saying for you is this is the resultant of the average of these two bands of the 3. Okay, resultant of the 3 bands of the 3. That is the reason why we choose an option C in this 1 and 3 are there. Now we are coming for the question number 3 discussion. Okay, we discussed about 2 past papers, MCQs. This is a subjective question. In this subjective question, they give the option to discuss about lack of prawn. This lack of prawn question, 29th question, always we all know very well, it is a case-based study question, structured question. In this question, they will be giving the comprehension or a paragraph followed by the questions of various types, which will be answered in this way, point by point, right? We all know very well. The lack of prawn is a concept which denotes the cluster of genes. What is this? The group of genes. So what we need to understand is group of genes are very important for controlling the other group of genes. So one gene is controlling the other genes. So this what is one gene denotes? So for example, let us consider if you're talking about one gene is making the DNA. Okay, that DNA is making the RNA. So this gene is controlled by the other set of gene. This gene is controlled by the other set of gene. This gene we used to call as, so for example, A gene is becoming A DNA is becoming A RNA. So whereas this gene A itself under control, itself under the control of gene B. The gene B controls gene A, right? So certain group of genes expression is controlled by the other group of genes, okay, in a different order depending upon the different parameters. For example, I'm a bacteria. Sometime I'm having the glucose nutrients. Sometime I'll be not having glucose nutrients. I need to sustain only the lactose nutrients. Sometime I'll be not having glucose and lactose nutrients. I'll be having only fructose nutrients. Accordingly, I'll be changing my metabolic pathway. Accordingly, I'll be changing my metabolic pathway. There is information you need to understand, right? In the case of question 3. So now, um, we know very well, I'm a bacteria. Let us consider this bacteria. When it is there with the glucose, lac operon will be negative. Lac operon will be negative. So if the bacteria is having glucose, so here, if we're talking about the glucose is there much more, lac operon is switched off. What do you mean about lac operon? The cluster of genes which are determining the lactose metabolism. This is the way the lactose metabolism should supposed to happen. Is controlled by a cluster of genes. Those cluster of genes we used to call as lac operon, right? Again, back I'm repeating, lac operon are also called as cluster of genes, okay, which are responsible for controlling the lactose metabolism. We are coming for the, if the bacteria is having less amount of glucose, and or zero amount of glucose, only lactose is available. So to sustain this bacteria will be using lactose. Again, back I'm repeating, bacteria uses lactose only in the absence of glucose, whereas in the presence of glucose, lactose will be not used by the bacteria. In the presence of glucose, lactose will be not used by the bacteria. This is the message we need to understand. The entire essence of this particular question. Right now, we are coming for the next phase of explanation. So, as I already told to you, when we are giving the explanation, everything should be written as point by point. So, not like a big paragraph, and then we need to give a lot of subheadings for the same. Right? As for the question pattern, option A, option B, option C question will be asked n number of time after giving a paragraph question. So for this paragraph question, the answer will be like this. For the diagram question, the answer will be like this. Or you getting my point? Okay. Now we are coming for the fourth question. In the fourth question, again back we took it from 2023, set 1, paper 1, question number 31. Matthew Meslin and Frederick Stahl experiment. We all know very well this Matthew and Meslin and Frederick Stahl are very important for carrying out what exactly? The DNA synthesis specification. For understanding the DNA synthesis specification, what we are doing is we are choosing the Matthew Meslin experiment. The second question not belongs to the molecular base of inheritance. The second question belongs to principle of inheritance. So they are having the probability of asking two different questions or the two different chapter in the question number 31. The first question belongs to molecular base of inheritance. Second question belongs to principle of inheritance. Whatever you need to avoid, you can avoid them, right? Now, based on the DNA replication process. So we are saying semi-conservative mode of replication. So this is double-stranded DNA. This double-stranded DNA, after getting segregation into two single-stranded DNA, this double-stranded DNA, after getting segregation into two single-stranded DNA, this single-stranded DNA is going to make a double-stranded DNA. This is the way the DNA replication happens in the eukaryote. So again, I'm repeating, the two DNA will move away from each other, and by using the two DNA or by using the two single-stranded DNA, we are getting the mature DNA, This is the mature DNA. Again, back I'm repeating, this is the mature DNA which is formed, or the daughter DNA which is formed. So this mature DNA or parental DNA, like this, we are not saying the mature DNA, the newly formed DNA is called what? Daughter DNA. Newly formed DNA is called what? Daughter DNA. Okay, the newly formed DNA is also called as daughter DNA. Are you getting my point up? So now we discussed this many number of questions in the molecular base of inheritance. We are entering into the second subtopic of this particular chapter, which is nothing but packing of the DNA helix. How the DNA is complicatedly packed or it is arranged very compactly means in the form of histone proteins. In the form of histone proteins. Or you getting me? So what you need to understand is basically histone proteins are the packing proteins. Again, I'm repeating. Histone proteins are the packing proteins, and non-histone proteins are also the packing proteins. If you're talking about histone proteins are basic in nature, whereas non-histone proteins are acidic in nature. The acidic non-histone and basic histone, acidic histone and what? Acidic non-histone are responsible for packing the DNA over each other. The DNA is packed over each other. The DNA is packed over each other because of the histone and non-histone protein. Because the histone and non-histone protein, the DNA is packed into a structure called, we used to call as a nucleosome. Nucleosome. Nucleosome is the message we need to understand. Now we are coming for the second phase explanation. The molecular base of inheritance. Can you see this nucleosome structure? So with your permission, just I'll take the antifungal spray. We're talking about the length of the DNA double helix in mammal. Basically, is having a 6.6 into 10 to the power of 9 base pair into 0.34 into 10 to the power of 9 base pair length. Okay, this length is much more than the typical nucleus. Here, typical cell nucleus, how much length of DNA it is having? More than that, who is having? The mammalian cell is having the DNA, right? So this type of packing we are not observing in the case of prokaryote. Okay, so the DNA is wrapped over the histone and non-histone proteins. The histone proteins and non-histone proteins are called packing proteins. Okay, and then I told which is acidic, which is basic, right? And this we used to call as octomeric structure. This octomeric structure, the word I am saying only for the what nucleosome structure. Okay, sometime in the CBSE board exam they will say octomeric protein, where you are observing this octomeric protein. We are observing the case of what histone and non-histone protein aggregate. Histone and non-histone protein aggregate will make the DNA to get packed and makes the DNA to get arranged in a compact nucleus structure. So in the compact nucleus structure, okay, this particular DNA is packed with the help of histone and non-histone proteins, right? So now we are coming for the next phase explanation is be very careful. So now in the prokaryotes, packing in the prokaryote. So packing in the prokaryote like this, whenever you're looking, eukaryote have a more intricate packing mechanism. Positively charged basic protein. Histones are rich in lysine and arginine. So lysine and arginine carries positive charge, indirectly speaks about basic amino acid. So you should be very, very smart. Okay, smartness comes because of your very good learning credential and also remembering credential. For example, when I was like you, so even though I am reading, okay, I used to revise the same content next day morning so that whatever I left in memory that I can recap while having a second time revision on the next day morning, right? So the basic amino acid lysine and arginine, which is positive, holding the negative charge DNA into a histone structure, which is making the nucleosome structure. Again, back I'm repeating, negative DNA and positive basic amino acid like lysine and arginine makes the DNA to get wrapped over each other. As a result, you are going to get an important structure called nucleosome structure, which is very, very important in the case of eukaryote, right? Now we are coming for the next basic message, in the DNA, whenever it need to participate in the DNA replication process, transcription process, the DNA will be getting unwinded away from the nucleosome structure. So unwinding of the DNA, again back I'm repeating, unwinding of the DNA. So unwinding of the DNA is possible. Unwinding of the DNA is possible. Okay. Possible in the sense the protein will move away from the DNA. Then only the DNA can go for efficient replication. Then only the DNA can participate in the effective RNA synthesis process. Like this, we need to understand the message. Now we are coming for the next basic segment in the packing of the eukaryote. The number of nucleosome in a mammalian cell. Number of nucleosome in the mammalian cell or any number. We know very well the theoretical number of nucleosome in the mammalian cell is extremely higher given the extensive length of the DNA. Right? The bead of the string structure of the nucleosome creates a highly compactly yet organized nucleic acid structure. Right? So there is a structure we already saw in the last slide. So how the DNA is compactly packed, and for packing this, we required histone and non-histone protein, and one nucleosome, another nucleosome, or interconnected by the nucleosome or interconnected by the DNA. One nucleosome, another nucleosome, are interconnected by the DNA structure, right? So nucleosome is nothing but a structure which is having protein and DNA. This structure we are not observing in the case of bacteria, virus, okay, and even in most of the fungus, right? So this type of structure we can see only in the plants and the animals, right? So now we are coming for the next major segment, the chromosomal function. Chromatin fiber packages nucleosome structure. So chromatin condenses to form the visible chromosome. Okay. Chromosome condenses to form the visible chromosome. Additional protein non-histone chromosomes aids in the high level packing material. This is a message. So in this, a lot of hidden concepts are there. So chromosome condenses to form visible chromosome during metaphase. Why? Why? Then only the chromosome splitting happens. So the chromatin condenses to form visible chromosome in the metaphase starting period. That means early metaphase. The early metaphase is a part of the cell division. In this early metaphase, what happens exactly? The cell is getting divided. Before the cell division, cytokinesis, okay, which should be completed? The early prophase should be completed. During the early prophase, the chromatin is getting condensed into chromosome. Then only, so because of this, what is the outcome? What is the outcome? They are not given in the slide. I need to explain. We are not here for reading the slide. We are here only for bringing a lot of hidden sentence behind each and every sentence of the slide, right? So we know very well the packed DNA need to move away from the histone proteins and non-histone proteins so that you are getting the DNA availability for the complete replication process and complete replication, transcription, and translation. Right? So now, what is the function of the euchromatin? So lightly packed up. What do you mean lightly packed up? Okay, here we can say tightly condensed. If you're talking about the tightly condensed DNA, the word I am saying, or highly coiled DNA, the word I am saying only for the heterochromatin. Only I am saying for the heterochromatin, whereas euchromatin, the word we are saying for the DNA is having less condensed area. So highly condensed DNA is not highly vulnerable or prone for getting involved in the RNA synthesis process, right? So there is a reason why we are saying this particular packed DNA of the heterochromatin, it takes a lot of time to get participated in the replication process as well as transcription process. Okay, heterochromatin will not participate in the DNA and RNA synthesis very fast. Slowly it happens, right? Now we are coming for the genetic material in the DNA. Before coming to the genetic material in the DNA, we are going for the related videos. Related videos, whenever you have time, Just examine it. For each and every subtopic, we not given the question. At the end of the each and every subtopic, we given the question for your better understanding, right? So now, packing of the DNA. In the packing of the DNA, what we need to understand is the theoretical number of the nucleosome in the mammalian cell is extremely higher, gives the extensive length of the DNA. Yes, of course. Okay, and the second thing is during metaphase, the chromatin condenses to form the chromosome. These are things we discussed.